Welcome to Cell Biology. Today, we are going to explore the fascinating world of the cytoskeleton. If you have ever wondered how a cell maintains its shape, moves around, or divides without collapsing, you are in the right place. We will build our understanding from the ground up, looking at the microscopic structures that make life possible. Let us start by imagining a bustling modern city. If a cell were that city, the cytoskeleton would be the steel girders holding the buildings up, the roads for delivery trucks, and the cables anchoring everything. Without it, a cell would just be a floppy bag of fluid. But unlike the permanent bones in our own bodies, the cytoskeleton is a highly dynamic, three-dimensional network of protein fibers. It can be built up and torn down in mere seconds, allowing the cell to adapt, crawl, and divide. Now that we understand what the cytoskeleton does broadly, let us look at its thinnest components, microfilaments. Measuring just 7 nanometers in diameter, you can think of them as a twisted pair of beaded necklaces. Each bead is a globular protein called G-actin, which snaps together to form a long filament known as F-actin. This filament has a distinct structural polarity, meaning it has a plus end that grows rapidly and a minus end that grows slowly. This polarity is absolutely crucial because it gives direction to motor proteins like myosin, which generate mechanical force. This dynamic building process leads us to the mathematics of how fast these filaments grow. Conceptually, the net growth rate of a filament is simply the rate at which new pieces are added minus the rate at which old pieces fall off. In this formula, the term on the left represents the change in polymer mass over time. The symbol K-on is the association rate constant, measuring how quickly new actin monomers bind. M is the concentration of free actin available in the cell. Finally, K-off is the dissociation rate constant, showing how fast monomers leave. If the free monomer concentration increases, the filament grows faster. Building on that growth rate, we reach a very important equilibrium concept called the critical concentration, denoted as CC. Conceptually, this is the exact sweet spot where the rate of monomers attaching perfectly equals the rate of monomers falling off. At this specific concentration, the filament neither grows nor shrinks. Its length remains perfectly stable. The formula shows that the critical concentration is simply the ratio of the dissociation rate, K-off, to the association rate, K-on. If the actual monomer concentration in the cell rises above this critical value, the filament will grow. If it drops below, the filament will shrink. Before we move on, let us see this with a concrete example. Imagine a researcher is studying actin in a test tube. We are given an association rate constant of 12 million per molar per second and a dissociation rate of 1.4 per second. We need to find the critical concentration required to keep the filament length steady. We choose our critical concentration formula because it directly relates these two rates to the equilibrium molarity. By dividing 1.4 by 12 million, the per second units cancel out, leaving us with molarity. The final answer is 0.116 micromolar. Physically, this means maintaining exactly this concentration keeps the actin filament perfectly stable. Now that we have mastered the flexible microfilaments, let us move on to the thickest fibers of the cytoskeleton, microtubules. If actin filaments are the flexible ropes of the cell, microtubules are the heavy-duty steel pipes. Measuring 25 nanometers in diameter, they are rigid, hollow cylinders built from alternating alpha and beta tubulin proteins. 13 linear chains of these proteins wrap around to form the hollow tube. Just like actin, microtubules are highly dynamic. They rapidly alternate between steady growth and catastrophic shrinkage, a behavior called dynamic instability, which is powered by the energy molecule GTP. This unique hollow structure gives microtubules their most important physical property: mechanical rigidity. They act as the primary support beams that keep the cell from collapsing inward under pressure. Beyond just support, they serve as a complex internal railway system. Specialized motor proteins act like tiny trains, walking along the microtubules to transport vesicles and organelles across the cell. Furthermore, during cell division, microtubules form a massive machine called the mitotic spindle. This spindle physically pushes the cell poles apart and pulls heavy chromosomes to opposite sides, a task that requires immense resistance to bending. To understand exactly how much pushing force these microtubules can handle before they buckle, we borrow a concept from structural engineering: Euler's buckling formula. Conceptually, this tells us the maximum compressive load a tube can take before it bends and fails. In this equation, F is the critical buckling force, measured in newtons. E represents Young's modulus, which is the intrinsic stiffness of the tubulin protein. I is the area moment of inertia, accounting for the hollow tubular shape. Finally, L is the length of the microtubule. Notice that because length is squared in the denominator, a microtubule's strength drops drastically as it gets longer. Let us apply this formula to a real biological scenario. A biophysicist isolates a microtubule that is 10 micrometers long with a known flexural rigidity of 2.2 times 10 to the negative 23. We are asked to find the maximum compressive force it can withstand. We use Euler's formula because it directly calculates this failure point. We substitute pi squared and the flexural rigidity into the numerator and square the 10 micrometer length in the denominator. Squaring the length is critical here, showing how rapidly stability decreases. Dividing the terms gives us a final answer of 2.17 piconewtons. Physically, this means a single microtubule can push with about 2 piconewtons of force before it structurally fails. Having explored the flexible actin and the rigid microtubules, we finally arrive at the intermediate filaments. Measuring about 10 nanometers in diameter, they sit right between the other two in size. You can think of them as thick, braided bungee cords or heavy-duty steel cables. Unlike actin and microtubules, which are constantly being built and destroyed, intermediate filaments are much more permanent. They're made of elongated fibrous proteins that twist together into rope-like structures. Because they do not bind energy molecules like ATP and lack structural polarity, their primary job is not movement, but rather helping the cell survive severe physical stress. This rope-like structure gives intermediate filaments their biological superpower: exceptional tensile strength. When you stretch your skin, the cells do not tear apart. This is because intermediate filaments inside them act like elastic cables, absorbing the mechanical tension. For example, keratin filaments anchor to specialized junctions called desmosomes, physically linking the cytoskeletons of neighboring cells into a continuous, tear-resistant sheet. If a person has a mutation in these keratin genes, even slight friction causes severe skin blistering. Additionally, a specialized meshwork of these filaments forms the nuclear lamina, creating a protective cage around the cell's DNA to prevent it from being crushed. Now that we have covered all three components, Let us look at them side by side in this diagram. On the far left, you can see the microfilament, made of actin. Notice its thin, twisted, beaded necklace structure, measuring just 7 nanometers across. In the middle is the intermediate filament. You can clearly see how the fibrous proteins twist together to form a dense, rope-like cable, 10 nanometers in diameter, perfect for absorbing tension. Finally, on the right, we have the massive microtubule. At 25 nanometers wide, its hollow, cylindrical structure, built from tubulin dimers, is perfectly engineered to resist compression, just like a steel pipe. We have covered a lot of ground today, so let us reinforce the most important ideas. First, the cytoskeleton is not a static skeleton, but a highly dynamic architectural system that gives cells their shape, strength, and ability to move. Second, microfilaments act as the flexible edges that allow the cell to crawl. Third, microtubules serve as the rigid steel pipes that resist compression and provide a vast railway network for transport. Finally, intermediate filaments function as the heavy-duty bungee cords that absorb mechanical stress. Together, these 3 networks collaborate seamlessly to keep the cell alive and functioning. Great job following along.